Welcome back to the lecture series on bioinformatics from CEC Gurukul lectures. And in the earlier lecture we talked about the genomics and how genomics has evolved from genetics and how important it is to study the genomics, that it can solve a lot of our problems. And we also kept a focus that because of rapid change and quick genome sequencing services, that it has been now possible to bring this into practice, and we have reached to the level of personal genomes, and where it has been extensively now applied in the medical and agriculture environment, all the fields. So we come now to the next type of omics, that is proteomics. As the name suggests, here we talk about the proteomes. And what is proteome? Proteome is the all the proteins of an organism that are expressed, meaning what all proteins are present in an organism is proteome. That refers to the proteome, and their study is proteomics. And like the DNA, each organism has its own unique proteome, because proteome is coming from the instructions which it has taken from the genome. So each species has its unique proteome, and that carries important information. So the interesting thing about proteome is that it is not constant. Of course, it took its information from DNA, but it is not constant, first of all, and it meaning it can be studied in the context of an organism or maybe a biological context. If I am studying all the liver proteome, maybe all the proteins in liver, I would say I am talking about liver proteome. So proteome is not constant, it differs from cell to cell and changes over time. What does that mean? I told you the gene, like I said, all the cells in human body, they have 23 pairs of chromosomes and they contain the same genes, and the point is that not all the genes are expressed at the same in the same cells. There is a differentiation in the expression, but proteome refers to the expressed genes, and so they will vary certainly from cell to cell, from time to time, in healthy and sick conditions. So proteomes carry a lot of information. So in proteomics we study when and where proteins are expressed, meaning I said just the example of liver. First we know where these proteins are expressed in an individual and when they are expressed, like certain proteins they are expressed when a woman is pregnant. So we have to know when and where these proteins are expressed. Similarly, the rates of the protein production, that is, they are being produced, and is it that they are being degraded at the same time, or what is the steady state of their abundance. Then how proteins are modified, for example, what are the post-translational changes which are taking place in these proteins. And it is also quite important to know what is the movement of proteins between the several subcellular components. The involvement of proteins in several metabolic pathways makes it very important to study them, because lots of diseases which we have are because of changes in the pathways or obstructions in the pathways. And we also need to study the proteins interaction among each other, how these proteins interact with each other to make a change in the phenotype or in any character in healthy or diseased state. There are several techniques with which we study these proteins in an individual at the large scale, and the most common is mass spectrophotometry based techniques, like tandem MS is very popular, or the techniques may be gel based techniques also, like differential in gel electrophoresis. These techniques, studying these techniques, huge amount of data has been gathered, and these data again requires proper storage as protein databases and taking out meaning out of this information contained in these databases. One is UniProt, that is UniProt knowledge base database that contains all the information about the proteins, and the focus here is on the function, meaning which protein is doing which function. Say for example, ATPase synthetase, it is synthesizing ATPs. So in this knowledge base we have protein sequences and the function related to them. Then we have INACT database. This database contains the all information about protein interactions. We have Reactome database, and that contains the role of the proteins in different pathways and processes. Here we talk about the reactions they've been carried out by these proteins. Then we have another important database for proteomic studies is the PRIDE database that contains the experimental evidence of published proteins and peptide identification. So here we keep only that information which is experimentally proved, and we talk about the protein identification and peptide identification. So this was about proteome, and the proteome and genome, I told you already, they are linked to each other. The information which is coming in proteins is in a way deciphered from the genomes. So both proteome and genomics, they have applications in medicine and agriculture, environment, everywhere. I will just quickly like to summarize in brief these application areas, and then we'll reach to transcriptomics. So these genomics and proteomic information have huge application in medicine. Like the first example is personalized medicine. If I can get my genomic sequence and proteomics information with me, I can be given personalized medicine, meaning the medicine which is very suitable for curing me especially, not for everyone. So a specialized personalized medicine is possible with this one. Similarly, we could have biomarker discovery with these informations, like a type of cancer, brain cancer, that is glioblastoma, and that is mainly gene expression. These genes are expressed. So for the diagnosis, if we check the expression of these genes and how much they are expressed, we can evaluate the patient's response for this brain tumor. So application here in biomarker discovery, the glioblastoma becomes one of the typical example. Then it is also used in drug discovery and development. How? Because when we know a protein responsible for this and we understand the protein structure, we can always understand and study in detail that which can be the potential drug targets and how these drugs are going to interact with these proteins and how much it can serve as a candidate therapeutic protein. So this information is really has come up with the proteomic studies. And similarly, system biology is that we can have system wide information about maybe the disease pathways and host pathogenic interaction, and when we study both of them together, the genome of the pathogen and the genome of the host, and that reveals very, very important and interesting information which is of huge application. Similarly, in agriculture, if we talk about the plant pathogen interactions, if we know, we can solve several questions in agriculture. And like this stress related environmental stresses, stress related genes and crop engineering, they have really been helped by these information coming from genetic and proteomic studies. Similarly, in the food science, food safety, quality control, allergen detection has really been improved with these tools. We have in paleoproteomics, what is happening is we can very more effectively study the evolution by comparative genomics and comparing the proteins, and we can know a lot of more information about archaeology. Similarly, astrobiology, what is astrobiology is when we study about the prebiotic organic matter and the exomicrobes, and that can be very effectively done by the sequences which we know. Once we know the sequences in here, we can study and compare and designing new systems also is possible when we know the genes responsible and the expressed product of these genes. So back to the same cartoon from where we started, is that now we know that it is possible, understanding that this searching your own name in Google search engine is controlled by a gene, because if this phenotype is present that you have this tendency, it is always possible to trace back this to on which chromosome, which gene it is located. And of course, with going further in the lecture, we will know how this can be improved or checked, or what exactly can be done about this. So here are some resources if you want to go for further reading here. This is a book on bioinformatics written by me, and there is another Bible of bioinformatics that is by Mount. I think all this information is very nicely given in these two books. So coming to the next part of this is transcriptomics. So what is transcriptomics? I already defined to you what is a transcript. Transcript is the product RNA which is synthesized by information contained in the DNA, and that is with the help of the enzyme RNA polymerase. So transcriptomics is the study of transcriptomes, and transcriptome is the complete set of RNAs transcribed in a genomic DNA. What does that mean? So transcriptome includes all the RNA in your body, and once we know the transcriptome and when we know the phenotypes, it is easy to identify that which RNA of this is being expressed. So the RNA which is expressed as protein, we call that as coding RNA, and the RNA which is not expressed, which is mainly regulatory in the function or which is helping in several pathways, that are non-coding RNAs. And the interesting information which is coming here is that if we isolate all the RNA in any system, we know that it is only 1 to 3% which is mRNA of the total RNA isolated from a organism, and I am talking of human beings once again. So some important facts or some interesting facts about these gene expression is that not all the genes are expressed at one time, meaning we have 20,000 genes, but at one time one set of gene is expressed. Here in our cartoon we were talking about that anxiety of searching your name. So when you are in anxiety, a different set of genes is expressed. Not all the genes are expressed at all the time, at the same time. Similarly, gene express depends on the cell or the tissue type. I hope you agree with this, because in my skin color, the color genes will be expressed, but in my heart, the other genes would be expressed. So not gene expression depends on the cell and the tissue type, and it also depends on the stage of the organism development and the stage of your life. Same way I gave you the example of pregnancy, a pregnant woman would have different genes expressed than a non-pregnant woman. So it also depends on health, disease and the environment, that how the genes are expressed, because again the same cartoon, it is the state of anxiety which is leading to the expression of those genes which are provoking that individual to search their name again and again. So it is very interesting to know about these expression of genes, and it is more interesting to know how this is regulated, meaning how it is decided that which genes are being expressed or which genes are actually expressing and leading to a phenotype. So the study of transcriptomics are very, very interesting and very, very important, because another important thing which is coming out here is that mRNA are not necessarily the sequences which have come from genes. Why? Because in the genes there are two type of regions. It's a sequence, DNA sequence, where we have introns and exons. The exons are expressed and retains for protein synthesis, but introns are spliced off. So when we see mRNA, which is working as a template for protein synthesis, this does not have introns. So the mRNA, when we isolate mRNA in transcriptomic studies, so we are talking about the expressed genes directly. So it becomes a tool to study the expressed genes, and in the vast human genome where we have so many genes to consider, and when we have so many pathways going on, if we could restrict ourselves by isolating this mRNA only to those genes where we have the genes which are expressed, it has really facilitated and it has really made things easy for scientists to express this in terms of phenotypes. So we can study gene expression, either single genes that we used to do in classical genetics, but here we do it globally, meaning we consider many genes together at one go, and we talk about variation in quantitative relative expression of many genes in global gene expression. So in transcriptomics we study global gene expression, and I hope you can believe me that it's an exceptionally powerful tool both in agriculture, environment and medicinal studies. So I told you already that gene identification in the systems are very complex if we talk about the eukaryotic systems. But when we study mRNA or transcriptomes or the coding sequences, if we isolate them, we could easily skip the introns, the genes which are not, the regions which are not expressed, or maybe the junk DNA. I told you already 99% of the DNA present in our body is junk DNA. That means they do not carry any gene or a phenotype. So transcriptomics has really brought a revolution that way, that we can identify which gene is responsible for which phenotype, and this way it has made it easy to study the response to a treatment or maybe to a cell population when we compare the mRNA isolated from one organism and then to the another organism in different type of stresses. We can know which gene is being altered in this stress. So it also allows the identification of retroposones. Now what are these retroposones? I hope everybody knows there is a reverse transcription going on from mRNA to back. They are synthesized and they are jumping, they keep moving in the genomes. So if we had restricted ourselves to the genes, we would not have known about these retroposones. So when we do transcriptomic studies, we could know about these retroposones also. Another interesting thing about transcriptomics is that it does not require any reference genome. Even if we don't have a reference data, still we can take out meaning out of this transcriptome in the transcriptomes. So this non-targeted nature of transcriptomics, this allow us the identification of new and novel networks in a cell. So not only the identification of gene which is carrying which phenotype, it is easy to identify the new pathways coming out if we know that there is particular mRNA present, and we can understand much more than we could have done only with the genomics. Apart from all this, transcriptomics provide us very important information about the gene regulation, because that way we know from where the gene is starting and where it is ending, and we know that at several times it is used to uncover the alternative promoters or maybe the novel splicing alterations which happens. So it gives a very important information, or it reveals the hidden or the complex pathways happening in our cells. So this is very interesting and unique, so that it simplifies the complex systems. The disease variants, what does that mean? Disease variants is, we can always compare a healthy individual to the diseased one, and we can know this by SNPs, that is single nucleotide polymorphism, or maybe the allelic specific expression, gene fusion. These techniques in transcriptomics have really facilitated the identifying the variant which is coming as a result of disease. Then immune related diseases are also sorted out, and we derive important information once we have the transcriptomes from, and we study the sequences which are coding these immune cell populations. So that was about transcriptomics, and I just considered the mRNAs which are transcribed. So once we have the mRNAs, we can know which genes are transcribed and which phenotype or which character they are controlling. Till now we talked about in transcriptomics the mRNAs, I said, but there is another interesting phenomenon in transcriptomics, is that the mRNA is unstable inside a cell, meaning we cannot retain mRNA as a mRNA. We need to do what? We need to synthesize a complementary DNA from this, and complementary DNA is what? Is complementary to this, and that it makes a double helical structure, but it becomes easy to store that double helical structure. Similarly, we have ESTs, that is express sequence targets, that help us in identifying the coding DNA in a cell. There have been several techniques with the help of which we could know the genes expressed at one stage. Like one example of such technique is microarray technology. We have talked about this in another lecture, previous lectures on gene expression technology. Maybe you can go back there and study how powerful DNA microarray technology is to understand the transcriptomics and the gene expression. So we have huge applications of transcriptomics, and we can identify the coding and non-coding regions in here. So we come to the last type of omics, that is metabolomics. What we do in metabolomics is we talk about metabolomes. Now what is metabolomes? Metabolomes represent the complete set of metabolites, that is small molecules less than 1.5 kilodalton in size, and they are metabolic intermediates or maybe hormones or signal molecules or maybe the secondary metabolites. They are very important in a biological cell or maybe a tissue organ, and they basically control, or they are the end products of several cellular processes. So metabolic profiling can actually tell you the physiological state of an organism. If we know all the metabolites present in one organism at one stage, so it is a snapshot of the physiology of that cell, and we can know what is happening and how do we control that. There are several databases for this. I will just cite one example here, that is Metlin. It's a database which contains the metabolites information, and it has 4,50,000 metabolites stored information about these metabolites. We have another database, that is the Human Metabolome database, and this also contains the metabolites information about several other databases. Then I just want to show you this picture where I am showing you that with the study of metabolites and transcriptomes, we could simplify these complex pathways and we can know what is happening. So again, to the same cartoon from where I started. By the end of this, I can tell you is that it is easy to sort out which gene is related with this phenotype and which chemical, which metabolite, which part of the plant body is affected by the stress which is induced and how it is expressed. So this was here, of course, because I took 4 topics in one go, that is 4 types of omics, but still I could convince you that all these four are related to each other and they are providing you invaluable information about the human organism, not only human, about many other organisms, and I gave you the numbers, the genomes which are accumulating, transcriptomes, and and several other omic studies which have come forward. So, with this one, I thank you for your interest in this topic, and I will look forward for any questions if you have. Thank you very much for your patient listening and interest in the area. Thank you.